Hey there. So we're going to start our review notes for our DNA unit. So the first thing I did is I grabbed a blank sheet of paper from my packet and I cut it in half because I only need half of it. And we're just going to sketch or draw out the process of protein synthesis. So I've actually got a few different colors of pens that I'm going to use. I've got black, red, blue, and green. So those are the colors that I'm going to use as we go through this illustration. So I'm start with my black pen. So everything starts here in the nucleus, right? That is where DNA lives. So I'm just going to draw my double helix. Let me zoom in just a smidgen. There we go. So, right, and I've got my nucleotides with my base pairs. So here's my DNA. And we know that DNA undergoes transcription to create mRNA. Right? So DNA undergoes transcription to make mRNA because DNA is stuck in the nucleus. It cannot leave, but mRNA can leave the nucleus in order to take that genetic message out to a ribosome so that we can make our protein. So now that we've undergone transcription to make mRNA, let's head out into the cytoplasm where we will find a ribosome. So this is my ribosome. And once I have found my ribosome, I'm going to redraw my mRNA, right? And we know that mRNA, right, has codons made up of the A, U, G, and C letters. So I'm just going to quickly kind of come up with a few codons, right? I'm making this up off the top of my head, but let's say we've got A, C, G, G, C, A, C, C, G, U, U, C, right? So on and so forth, right? However long the gene is. But now I've got my first codon, right? Every 3 letters is a codon. And so now that RNA has the first codon, it will read that codon and it will look for the corresponding tRNA. So this is our tRNA, right? We know T stands for transfer. And the tRNA, right, has the amino acid that we need because amino acids make proteins. And it also has, let's say that, it also has the anti-codon. And if the anti-codon matches with the codon, then the ribosome will drag it in and start putting them together. So let's draw a few more tRNAs. Do another one. Yeah, we'll do one right here. There we go. All right, so that's my amino acid. Here's my anticodon. So if my codon is A, C, G, my anticodon would need to be U, G, C. Right, that's my base pair rule. A would pair with U, C would pair with G, G would pair with C. So then this tRNA would come here, U, G, C, right? The codon matches the anticodon. A little peptide bond would form. And now we need the next anticodon to deliver the amino acid. So GCA would pair up with CGU. So now this tRNA will come CGU, right? And then we'll get the next one and the next one and so on and so forth, right? And all of these amino acids are being held together by a peptide bond. There we go. Now, with protein synthesis, or specifically with translation, right, in translation, we are going from mRNA to a protein, right? And this is occurring on a ribosome in the cytoplasm. We know that in order for translation to start, right, we have to have our start codon, right, AUG. I didn't really worry about that here in our example, but we know that, right, the first codon that the ribosome would need to read would be AUG in order to start the process of protein synthesis. And then, of course, we need a stop codon, right, which could be UAA, UGA, or UAG. So there's one start codon for protein synthesis, but 3 possible stop codons for protein synthesis. So in summary, protein synthesis, right, is DNA to mRNA to a protein. And DNA to mRNA is transcription, and mRNA to a protein is translation. So now that I have my illustration, move my pens out of the way, I'm going to place this in my notebook on page 60. I'm just going to turn my notebook sideways like this, and then I'm going to grab some tape. There it is. And I'm going to tape or glue this into my notebook on page 60. Okay, so now that I have taped or glued in my diagram here on page 60, let's take a look at page 61, sticking with our protein synthesis review notes. So I'm just going to make a couple columns here to review protein synthesis again. So here in this first one, I'm going to call this my original DNA. I'm just going to pull a line right there. And then resulting mRNA, my codons, and then my amino acids, and then my final product, which of course is my protein. All right, so I'm just going to continue these lines down just a little bit. So my original DNA is going to be, right, double-stranded. So T-A-A-T, let's just do one side. C-A-C-G-G-T-A-C-G-T. Oh, I was off that one. So now I can just do the other side with my base pair rule. C-T-A-T-G-C-A. Perfect. So here's my original DNA. I'm going to go ahead and just finish off my columns so I don't confuse myself on what I'm looking at. So my original DNA, right, when I'm moving from DNA to mRNA, right, I know that that process is transcription, right, transcription. So I'm going to take this side right here, and I'm going to use it, the left side, to create the resulting mRNA. So if I am, the enzyme to make mRNA, I see a T, I'm going to pair it with an A. See an A, I'm going to pair it with a U. C to G, A to U, C to G, G to C, G to C, T to A to U, C to G, G, C, T, A. So now I have my mRNA. So the codons, all right, that I actually have, remember codons are every 3 letters. So my first codon is A, U, G, right? My next codon, U, G, C, C, A, U, C, C, A. So I just wrote out my codons just to make it a little bit easier for me to see them so that when I start translation, right, to find my amino acids, I don't make a mistake. So I can now use my codons and using my codon chart, I can find my amino acids. So if my codon is AUG, right, I know that that gives me the amino acid MET. That's my start codon. So I'm going to say my first amino acid is MET. My next codon, UGC. UGC gives me CYS. CAU. CAU gives me HIS. And then CCA. CCA gives me, oh, I made a mistake here. This should be a G. Ooh, sorry, GCA. GCA gives me ALA. Right, so always double check ourselves so we don't make a mistake. So basically our final product, right, if this was a protein, right, MET, and then peptide bond connected to cis, peptide bond connected to his, peptide bond connected to ALA. So we finally have our protein. So DNA, right? Where is it contained? Right? DNA is contained in the nucleus. The resulting mRNA, where is it produced? Right, mRNA is produced in the nucleus. Right, mRNA has to be made in the nucleus because that's where DNA is. Our codons, where are they read? Right, they are read on a ribosome. Right, mRNA leaves the nucleus, heads to a ribosome. The ribosome reads it. Amino acids, where are they assembled? Right, they are assembled on a ribosome in the cytoplasm. So that's it for our notes here on page 61. Again, just showing the overall complete process of protein synthesis, transcription to translation, and then just answering a couple questions down here at the bottom, just to remind ourselves where all of this is occurring in our cells. All right, so for our last page of notes, I'm here on page 62. I have drawn a Venn diagram. So just 2 large circles. I've labeled the left one DNA, the right circle RNA, and then of course the section in the middle is going to be for both. And I'm just going to compare and contrast the differences and similarities between the two nucleic acids, DNA and RNA. So DNA, we know, is only found in the nucleus. We know that it contains all of the instructions for our traits, right, for our proteins. It has 4 bases, A, T, C, and G. DNA is double-stranded, and it has the sugar deoxyribose, right? Deoxyribose sugar. As for RNA, right, RNA can be found in the nucleus or the cytoplasm, right? If we're talking about mRNA, tRNA, and even rRNA, it also has 4 bases, but it has A, U, G, and C. RNA is single-stranded, right? It's a single helix, and it has ribose sugar. And then as far as what DNA and RNA share, right, they both have A, G, and C bases. They both carry genetic information. Right? RNA helps express our genetic information through protein synthesis, right? Translation. They are both found in all living things, right? In order to be living, you have to have genetic information. Humans, we happen to have both, right? DNA is our main source of genetic information, but we do have RNA to help express it. And that is it. So that's it for our Venn diagram. That's it for our DNA review notes. If you have any questions or you need to go back over any of these 3 pages, just let me know.